right, so grade 12s, we are going to jump into the work from genetics. Hopefully, you have started with this term with your teacher. So we're going to go through all the types of inheritance, and then we're going to end today's lesson by doing some practical examples. So hopefully, you are all with us so that you can follow us with the examples. All right.
Right, let's start grade 12. So we're going to start with the types of dominance, see, that you get. And there's, first of all, there is complete dominancy, and that means that one allele is dominant and the other one is recessive. Now, first of all, I'm going to just stay with this and look at the term, because there's a lot of new terms that you've learned or are going to learn in this session. And the first one is allele. So I want to quickly explain what is meant by the term allele. So I'm going to take you back to a chromosome, and I'm going to draw an unreplicated chromosome. So if you remember from grade 10 and then from Meiosis, your teacher taught you that a chromosome that consists out of only one leg we call an unreplicated chromosome. Now on this chromosome there's an area that we call a locus, and on that locus there's a gene. So the gene, in genetics, we use a symbol, different symbols to identify that specific gene. So say for instance I use the symbol R for that specific gene, whatever gene that might be. It's on a specific area which we call the locus. Now if you remember that chromosomes, they come in pairs, and the one is from the mother and the one is from the father. So if I draw the other pair, the other chromosome, on that area there will be the same gene as well. And we're going to use the same symbol for this example, but say for instance it's the same gene or the same allele, then we see that genes come in pairs, and this pairs we call then allele. So if we go back to our definition, we see that complete dominance, the one allele is dominant and the other one is recessive, which means that this dominant allele will mask the recessive allele in the heterozygous condition. So what is meant by the heterozygous condition? I need to go back to my whiteboard, and I'm going to look at these chromosomes that I've drawn here. And here you can see that both alleles look exactly the same. They're both capital letters. That means that it's homozygous condition, because both alleles look exactly the same. We can have it in the dominant condition, or if I redraw the chromosomes again and I draw the recessive alleles, remember chromosomes come always in pairs at a specific area which we call the locus. There might be two recessive alleles, which is then the small letter R. Now if these two chromosomes, one is now from the mother and one is from the father, if these two come together in the zygote, we're going to have once again two chromosomes, and then the one comes from the mother, that specific area, that's a capital R, and then on the other side the one comes from the father, which is then the small r. And this condition where the two alleles look different, we call then heterozygous condition. So once again, to the definition, we see then if one allele is dominant and the other one is recessive, such that the effect of the recessive allele is masked by the dominant allele. So if I look at this picture that you see here in front or on your slide, we see exactly what I've drawn now on the whiteboard as well. We've got a red flower and we've got a white flower. Both these parents, they are homozygous, so they look exactly the same. Their alleles, this red flower's allele is exactly the same, and the white one is also the same. So if we look at the offspring, we see that even though all the offsprings are red, their alleles aren't the same. They are heterozygous. And that's what is meant by complete dominance. We can't see that recessive allele. So all the flowers, they look red, even though they do have that white allele in their genotype. So that's the first definition that we've got. If we move to the next definition, which is incomplete dominance. Now, in incomplete dominance, neither one of the two alleles of the genes is dominant over the other one, resulting in an intermediate phenotype in the heterozygous condition. All right, so let's quickly unpack the definition here. If we look at the picture again, the phenotype is what you see. So I see here a red flower and a pink flower and a white flower. Now, how do I know that it's incomplete dominance? As soon as you see three different characteristics, I immediately know that it's incomplete dominance. I'm quickly going to jump back to the previous slide. Here we can see there's only two colors. There's either red or a white. You will not find a third characteristic. There's only two characteristics, and that is typically of your complete dominance. If I jump over to incomplete dominance, I can see that there's a third characteristic. Now, please don't get confused with paint colors where you think, okay, but it's red and white. If I mix the two, I'm going to get pink, and that's why it's incomplete dominance. Remember this, the one in the middle, your heterozygous phenotype, can be any color. It can be red and white with a yellow flower. It can be any color. The importance here is that you will have three characteristics. Now, if we've got incomplete dominance, the two alleles that you see here, the one for the red and the one for the white, the one can't dominate the other one, and that's why you find almost like a in-between between the two alleles, an intermediate phenotype, which is then in this case a pink condition. What is important for this type of dominance is that when you do the genotype, then we will use only capital letters, where in complete dominance we work with the same letter. The dominant characteristic is a capital letter and the recessive allele is a small letter, where in incomplete dominance you will only use capital letter. Remember, in the exams they will give you the letters. So as soon as you see that there's two capital letters, then you know that it's not complete dominance, it's most probably incomplete dominance, or it can be codominance. Now, what is meant by this one? Let's just quickly look at the definition. both alleles of the genes are equally dominant, whereby both alleles express themselves in the phenotype of the heterozygous condition. Alright, so what is meant by that? Once again, we've got a red flower and we've got a white flower. But in this case, this characteristic is dominant, the white one, as well as the red one. So we won't find an intermediate condition. We will have both these characteristics will be seen in your phenotype. And that's why this flower will have white petals as well as red ones. Both characteristics will be present in your complete dominance, or codominance.
All right, so with genetics, we can also, we are also going to look at a very important scientist, and his name was Gregor Mendel. All right, he is known as the father of genetics, and he came up with a few laws to, that sort of, in his words, explains genetics. So his three laws are the law of dominance, the law of segregation, or the principle of segregation, and then his third law is the law or the principle of independent assortment. Right, so I'm going to unpack these three laws for you. You will need to be able to give a description of these laws in the exam. So your teacher could ask you, describe Mendel's law of dominance, right? And then this would be sort of what you will be explaining. So I'm here to just explain what his law means. So his first law is the law of dominance, and he you'll see in your textbook that there are many peas, examples with peas, and he spent a lot of time looking at peas and did experiments with them, and he basically crossed a purebred, let's say, what example is this? A purebred long pea with a purebred short pea, right? And then he, after some time, the next generation, the offspring of the long pea and the short pea, made a new plant, and he realized that all of the offspring of the new generation, they were all tall peas, right? They were not short at all. So even though one parent was completely purebred short, all of the offspring, basically, he saw that all of them were tall. So that is what he observed, and he used that to come up with his law of dominance. So he said that when two homozygous organisms, so that is basically like a purebred tall and a purebred short pea, they are crossed, then all of the individuals of the next generation, and we'll explain what this F1 generation is in a bit, but all of their offspring will then display the dominant trait. Right, so because in my example all of the offspring were tall, therefore he said that being tall is then the dominant trait, or the dominant allele for this gene. They could mask the recessive trait, the short trait, and that could only come up in the next generation. But we will do a few examples with crosses so that you can basically see with examples how this law of dominance play out. So at the moment it feels a bit like all of the information are all over the place, but his first law of dominance is a basic in terms of genetics. Right, let's look at the second example. So this is his principle of segregation. So he says that an organism has two factors that is actually 2 alleles. So your exam guidelines uses the word factors. So every organism, you, me, we all have 2 factors for a certain gene. 2 alleles, forms of a gene. So if you have blue eyes, that gene for blue eyes, there are 2 factors that control your eye color. If I have one blue eye factor and another blue eye factor or an allele, then my eye color will be blue. So there's 2 factors that control these genes or the expression of these genes, and during meiosis these 2 factors are going to separate from one another, right? And that will then contribute to the factors in the gametes. And then his last principle, or his last law, is he said that these factors, these alleles, that they are not dependent on another allele. So just because, let's say that you have inherited the blue eye allele, it doesn't mean that you will then necessarily inherit the being tall allele. One allele isn't dependent on another allele. They're not going to influence each other, right? So that is his 3rd law. So I hope we are going to move into practically understanding genetics.
Right, so before we start with the cross, you're going to remember that during this section of the work, they're going to give you a lot of crosses that you need to do. And therefore, it's very important for you to remember the template. Now, what is nice about this template is if you do it, you will automatically get 2 points just by writing some of the information down. So let's quickly see, what do you need to remember? First of all, whenever you start to do a cross, you start by writing P1. Now, P1 means parent 1. So that's the first information that in a question they're going to give you. And what about the parents? So you write P1 down. Then after that, you need to give the phenotype and the genotype. Remember, the phenotype is what do you see? What about the characteristic? Like ma'am said, tall and short, or blue or brown eyes. That's what we see. Genotype is what is happening at the genes. What alleles does that specific individual have that you need to write down? Luckily for you, if we look at the different information, phenotype, it's the visible characteristics, and it's very important that you write the complete information. So you cannot only write red and white for the examples that I had. You have to write red flower, white flower. Then by the genotype, you write down the alleles, but luckily you will receive the symbols. They will give that to you in a question. In brackets, they will give you a capital or small letter O, whatever the example entails. After that, you write down meiosis, and then you give the gametes. I like to tell my learners, whenever you write the gametes, you draw a little circle around each gamete so that you will remember to write them separately and not all together, because you will lose marks. Then fertilization, please look out for this. Some textbooks, they talk about fusion, but you must write down fertilization. That's the word. So if we look at these 2 words, meiosis and fertilization, you will already, by writing down these 2 words, you will get 1 mark. Then you draw your little Punnett square with the gametes in the top row and in the first column, and you do your cross there. Then after that, you write down F1. Now the F1 is the first generation. How does the first generation of offsprings look? That's what's meant by the F1 symbol. And then once again, phenotype and genotype. But here's a very important thing that you need to remember. In your question, they're going to ask you either the ratio or the percentage of the phenotype or the genotype, one of those two. Then it's important that you give the answer. You cannot, if they ask percentage, then you give the ratio of the phenotype. Or if they ask the ratio and you give the percentage of the phenotype. So it's very important that you read what do they want from you. And then if you give a ratio, remember to give it as simple as possible. Not a 2:2 ratio, but a 1:1 ratio. So if I summarize what I've explained to you just now, it's then the monohybrid cross. What is meant by a monohybrid cross? That's a cross where we only look at one characteristic. Remember, there's a lot of characteristics. In this case, if we do genetics, we will only pay attention to one specific characteristic, which is meant then, or what is meant by monohybrid cross. Then the marks, you see, by writing P1, F1, you get 1 mark. By writing meiosis and fertilization, not fusion, fertilization, you get another mark. Then a lot of times there's a compulsory mark, and the compulsory mark will be somewhere here where they ask you give the ratio of the genotype for the F1 generation or the percentage of the phenotype of the F1 generation. So that's where the compulsory mark is allocated. And then there's other places. So you get a mark for phenotype there by P1, genotype, you get a mark for the gametes, and you get a mark for the genotype and the phenotype, depending on how they asked the question. The next terminology that you need to know is sex determination, to determine the sex. So when we look at that, once again you see on the side we've got that template, and we know that chromosomes of the humans, you get 22 pairs that are autosomes, and then 1 pair of chromosomes that we call the sex chromosomes, the gonosomes. Now in humans, males, they've got an X and a Y chromosome. In females, there's 2 X chromosomes. So if we look at this picture, we see there that the male only got 1 X chromosome and then the Y chromosome. Now what is important about the Y chromosome is that the Y chromosome is a lot smaller than the X chromosome. So Ms. Berger will explain to you just now sex-linked crossings, and then you will understand why that X chromosome is so important, due to the fact that the Y chromosome is much smaller. It's lacking some of the chromosome, or the genes, not the chromosomes, the genes. And a female, they've got double X chromosomes. So if we look at this cross, you can see that at the end, after a male and a female is crossed, you've got a 50% chance of getting a female and a 50% chance of getting a male. We use, if we arrange the chromosomes here, we call that a karyotype, and the karyotype is where we've arranged all the chromosomes according to their size and where the centromere lies. And luckily for you guys, you will receive a picture as it is. You don't have to arrange them. There's they're already arranged. What you need to do if you get a picture like that is to identify whether it's a male or a female. So if we go to the last chromosome pair 23, there you see there's the Y chromosome. It's much smaller than the X chromosome. So in this case, this karyotype is that of a male. And then the other thing that you need to be able to do is you need to go to chromosome pair 21 and identify whether there's a genetic defect, Down syndrome, which is not part of the work that we're going to do today, but that's where you use the karyotypes, is for those type of questions, to identify whether there's some sort of genetic disorder and whether it's a male or a female. They've arranged it there from 1 to 22 is your autosome, and then the last one is then your gonosomes. That is there.
Okay, so grade 12s. Next up, we're going to look at sex-linked inheritance. And as Ms. Schultz said, that with sex-linked inheritance, there's one pair of gonosomes. And we call these gonosomes could be an X, two X's, or it could be a Y, an X and a Y chromosome. And with sex-linked inheritance, that allele is only found on the X chromosome. So there are two disorders that we are going to look at that are sex-linked. One is color blindness and one is haemophilia. Before we have a look at that, I'm just quickly going to show you some of the formula or the formats that we'll use to explain the work. So if this is someone's two X chromosomes, then we're going to say that this person is a female. All right. If someone has an X and a Y chromosome, then that person would then be male. All right. So with sex-linked inheritance, the defect is only found on your X chromosome. So if this person has the defect and the defect is recessive, then this is a female that definitely has that condition. So in the exams, we're definitely not going to use little dots on a paper. We're going to put a letter on that X, right? So let's, for example, put a little H there and a little H there. So this will now be the genotype of a female with haemophilia. I'm just going to write a little H there. So this is a female with haemophilia. For a man to have haemophilia, he only has one X chromosome. He has a Y chromosome, but this condition, because it's sex-linked, this condition cannot affect the Y chromosome. Right, so because he only has one X, and let's say this man inherited the defective allele, then this means that this is a male with haemophilia. This is a man that is affected. If that man didn't receive that allele, then he would be perfectly healthy. Right, so because it's sex-linked, this condition is sex-linked, the condition is linked specifically to the X chromosome. Right, so you can see that I've written an extra letter on each one of the X chromosomes. Right, and we're writing it as a superscript at the top, not at the bottom, not big, it's sort of hanging at the top. That is the format in which you will have to give this in the exam. And you can note that there's nothing linked to the Y chromosome. Alright, so back to our notes. With sex, the two conditions that we'll look at is color blindness and haemophilia. And with color blindness, you just need to know a basic idea of what this is. So color blindness is a defect where men or women, they don't have the ability to distinguish between different colors. So if I put that in the same format on my whiteboard for you, so they will tell you in the exam that, let's say that is the allele for color blindness. Color blindness. Right. So an X with a little C at the top. If there's a female that is colorblind, right, that female's genotype, she's a female, so there should be 2 X's. She could have XC and another XC, and she is colorblind. Because she has 2, and colorblindness is a recessive condition, she could also only have the 1 allele and then nothing on the other X, right? Yeah, so there's only a capital C. Yeah. Actually, the first one should be the capital, and then the second one should be the little c. So she has a big C and a little c, and the little c is sort of hidden, masked by the big C. So this is a female that is affected. She has color blindness because she has both of the little c alleles. This is also a female that does not have color blindness. She's not affected, right? She is normal because the big C, the healthy not affected allele, overpowers the little c allele. In a man will be XY, right? If the man inherits the big C allele, the healthy C allele, he would be unaffected. If the man inherits the little c allele, the C is a bit of a difficult one to use, he will be affected. I think I'm just gonna add the defective one and put that in purple. Alright, so this female inherited both of the defective alleles, so she's affected. Here you can see this female only inherited one of the 2, and this sort of explains that law of dominance, where this person is heterozygous, but because the dominant allele masks the recessive allele, this person is not affected. Alright, then the next example that we'll use is haemophilia. Right, so you need to know what haemophilia is. So it is a condition where your blood does not clot. Right, you don't have the factors to make your blood clot. So someone that has haemophilia is also known as bleeders. Alright, and again, this allele is carried on the X chromosome and it is recessive. Alright, the next type of inheritance, or not really inheritance, but the next example that we'll look at is with blood groups. So with blood groups, you should know that there are 4 types of blood groups. So I'm going to add them here for you. We refer to them as now the phenotypes, how your blood group is expressed. Right, so there are 4 blood groups, and we're going to call them the 4 phenotypes for blood groups. They would be blood group A, blood group B, blood group AB, and blood group O. Alright, so on the board we have blood group A, B, AB, and O, and those 4, they are known as your phenotypes. Right, so to you inherited these blood groups, and if you are blood group A, then you could have inherited this allele, an A allele, so this is the notation that you will use, and you could have inherited another A allele. So you could have inherited the first one from mom, the second one from dad, and that combination gives you the phenotype blood group A. Right, and this combination over there, we call them your genotype. Right, looking at B, for blood group B, you could have inherited the B allele from mom, the B allele from dad, and that gives you blood group B. You are homozygous for B because you have two of the same alleles. For blood group AB, you inherited the A allele from mom or dad, and you inherited the B allele from the other parent. Right, that is the notation. Then for blood group O, this sort of brings about a new, this will influence the blood group A and the blood group B. For O, these two alleles are your recessive alleles. So they, you have inherited the recessive allele for O and the recessive allele for O, and that gives you this genotype. So here you can see that there's a dominant allele, the A's and the B's, they're written in capitals, and then there are recessive alleles. So that means if you're blood group A, you could also be one A allele, which is dominant, and then you could also inherit the O allele, which is recessive. So then this would be a heterozygous condition for blood group A. If you're blood group B, then you could also have inherited the dominant allele for B, as well as that recessive I. So this is a table that I would tell my learners, if I just go back to the slide here, this is an important table. This is the phenotypes. Here we have the genotypes, and you will actually have to go and study this table. You will need to use these genotypes to be able to answer genetic crosses. All right. So quite important that you study that, and also blood group AB is also where we can see codominance being played out, because there's an A allele and a B allele, and we can see that the phenotype actually expresses both of these alleles, that one allele doesn't overpower the other, so they are both expressed in the phenotype. Then just very shortly, a dihybrid cross. In a dihybrid cross, we are looking at two characteristics at the same time. So this is sort of the last thing that I do with my grade 12s, because the genetic crosses are just a bit longer. So there we'll look at someone, let's say someone that is, or a flower that is tall as well as purple, and we're going to cross it with a flower that is tall and purple. So what you can see is that there are two features, two characteristics that we are crossing at the same time. We're not only crossing a tall individual with a tall individual, or a purple and a purple. We're crossing a tall and purple with a tall and purple, and this type of cross, we call this a dihybrid cross. All right. The next line of work is called a pedigree diagram.
Right, so in the genetic pedigree diagram, all these crosses that we've done, they can ask it in a pedigree. Now, what is meant by a pedigree is this little diagram where they want to show you who married who and where the defect is seen. So if we look at this picture, what is important, the example that I give you is brown eyes and blue eyes. Now, if you get a pedigree or genetic pedigree, you have to go to this bottom part where the information is, your key. And then you see the white little block is the man with blue eyes, and the one that is colored is then a man with brown eyes. The circle is always meant by female, and that's a female with blue eyes and a female with brown eyes. So if we unpack it, we see that Joshua married Runel. Runel's got brown eyes and Joshua's got blue eyes. Now in the example, they tell you that blue eyes is dominant, blue eyes is recessive, and brown eyes is dominant. So what I normally tell my learners, if you get a diagram like this, it's very important to quickly go and write down all your recessive characteristics. So all the white circles and squares, go and fill in their genotypes, because you know for a person to have blue eyes, seeing that blue is the recessive trait, both the alleles must be small letter b. So if we go back to Joshua, the father, both his alleles must be small b, small b. We don't know what Runel's alleles is, both her alleles. We know that one of her alleles will be at least a capital B. So if I quickly go to the whiteboard and I redraw the first part, we've got Joshua and he's got blue eyes. That's why I say you write down the genotypes of the heterozygous recessive trait, because that you know will always be like that. And then the mother, which is Runel, we know that she is brown eyes, but for her to be brown eyes, she must have at least one dominant allele. But we don't know by just looking at the first part whether her second allele is a small letter, meaning she's got the recessive allele for blue eyes, or is maybe she is homozygous and she's got the capital letter, so she's got the brown allele as well, being homozygous dominant. We don't know. So we must go to their offsprings, and if we look at the offsprings, we see that they had two children. They've got a daughter and a son, and both their children, the circles is colored in. So that means that the daughter as well as the son has got brown eyes. Now for them to have brown eyes, let's just quickly go back to the father. Remember, each parent gives one allele. So if I stick to Joshua, the father, one of his alleles must go to each child. So say for instance, this allele goes to the daughter, that's a small b, and say the same one went to the son. For them to have brown eyes, it must be that the mom gave a capital B, the dominant allele, to both her children. Right, so if we go back to the slides, we see then that Joshua's got is homozygous recessive. Runel, we still don't know, because we can't see from the children whether she is homozygous or heterozygous. The horizontal line between the two squares is if they married. So Peter married Veronica, and Veronica's got blue eyes, and in their offsprings, they had a lot of children, 5 kids, we can see that there's children with blue eyes as well as children with brown eyes. For them to have children with blue eyes, we know that Peter must be heterozygous. So if I go quickly back to the whiteboard, and we see to demonstrate that to you, Peter, he married Veronica, and Veronica's got blue eyes. For her to be blue eye, she must be homozygous, and both her alleles must look the same. Now they had different children. I'm not going to redraw all the children, but for them to have children with blue eyes and children with brown eyes, I'm going to remind you again, remember each parent gives an allele. So Veronica, she's homozygous, so she gave the small b, and the small b goes to that child. For them to have children with blue eyes and brown eyes, the father must be heterozygous. For him to give a capital B for the one child, but he must also give a small b for another child, for him to have both brown-eyed children and blue-eyed children. So if I go back to the slides again, we see that when Peter married Veronica, they didn't have only kids with brown eyes, they had children with blue eyes as well. So Peter must be heterozygous for them to have kids with blue eyes. Mutations, that's definition. Please, grade 12 learners, you have to learn this definition as it stands here. It's very, very important because it's a common question that they ask quite often in the exams, and then the kids battle to explain it. So let's quickly see. A mutation is a sudden change in the genetic composition of an organism. We get 2 different types. In your textbooks, there might be more types of mutations. Luckily for you guys, you only have to learn these 2. So the first one is a gene mutation. And a gene mutation is the change in the sequence, very important, of the nitrogenous base of the nucleotides in the DNA. Now, normally they link this mutation to DNA coding of DNA. So if they ask questions with DNA replication, protein synthesis, normally this mutation is linked to that section of the work. The other type of mutation is chromosomal mutation, where there's a change in the normal structure or the number of chromosomes. So for a question like that, it's a lot of times it's linked to meiosis, and there you will get this karyotypes, and they want you to identify whether there's a chromosomal mutation. So you can just go and see if there aren't somewhere an extra chromosome or a chromosome that looks different from its partner. Genetic engineering. So for genetic engineering, we see that it is the manipulation or the transfer of genes from one organism to another to make products that satisfy the human needs. So for genetic engineering to happen, the humans want to have some characteristic. We need something in the industry. It might be in farming to have a resistant gene for specific trait, and then we will manipulate the organisms to get that specific gene, and then we call it genetic engineering. In genetic engineering, we've also got stem cells. Now, what is a stem cell? That's a little bit of definition that you've done in grade 10. That is cells that are undifferentiated and have the potential to develop into any type of cell to replace the affected or the defected cell, causing a disorder. Now remember, we can get stem cells from embryos, which normally has got some ethic problems, but we also can get stem cells from adults. So I can harvest your stem cells by drawing, for instance, red blood cells from your blood or from fat tissues. We can also get stem cells, which we can then use to replace this affected cells that might be caused by a disorder. Another important type of question is cloning. Once again, it's grade 10 work, and they quite often like to ask the question about cloning. So if we look at cloning, we see that there's an adult that we want to clone. So in this adult, this sheep, there's a specific characteristic that we as a farmer like. Maybe the wool quality is brilliant, so we want to have a lot of sheep with that specific characteristic. So then I will take cells from that sheep, not its egg cells. We will take, for instance, skin cells, because in that skin cells, in that nucleus, is that characteristic that I as a farmer want to have in my next generation. Then I will take another female sheep with characteristics that I don't really want. I will take an egg cell from that female, and I will remove the nucleus because I don't want that characteristic. That's not what I want. I want this specific sheep's characteristics. So I'll remove the nucleus of the one female, the donor female, and then I will put the nucleus of the adult or the sheep with this desired characteristic. I will put inside the egg cell, give it a little electrical shock so that cell division will take place, and then when there is an embryo, I will transfer this embryo into a surrogate mother. That's a different sheep again. It's not the donor or the sheep with the egg cell or the one with the characteristic, the desired characteristics. It's just a surrogate mother that will carry that lamb till it is born. So none of her characteristics is in that cloned animal. This cloned animal must be exactly the same as the sheep that I originally wanted. It must look exactly the same. So cloning, they like to ask, you must be able to explain this process. Very simple. And once again, a lot of times they ask you about the cells. What characteristic is in this cell? Why did you use skin cells and not the egg cell of the cloned sheep? It's of course because the skin cell already got the desired characteristics, where an egg cell isn't necessarily got that characteristics. Why did you remove the nucleus of, say for instance, adult B? Because it it's got the characteristics that is not desirable, which we don't need. Is there any characteristics of the surrogate mother in the cloned animal? And then the answer is, of course, no. Right, grade 12, I think we might have a quick time for a question. So let's just quickly have a look at a question where brown eyes sex-linked to dominant alleles on the X chromosome. So they ask you, explain. This is a very important question, and that's why I want to end with this question. Why more males than females will have white teeth? And if we look at this question, remember that the males, they've only got one X chromosome, and therefore they will inherit, if they inherit only one recessive allele, that characteristic will be visible in the phenotype, where females got two X chromosomes. So they have to inherit both recessive alleles for them to be able to have white teeth. Please go and learn this answer, and you must be able to apply it to any question that is to a sex link, because a lot of times they ask you to explain why a specific sex link trait is more common in males than females. Right, grade 12, I hope that we tried to help you with genetics. It's a very difficult topic, but go and work through your questions, ask your teachers for help, and I hope that you've got a good afternoon. Goodbye.